In this screencast, we are going to talk about DNA replication, which is the process where an exact copy of DNA molecule is produced. And then we are also going to introduce RNA as well. And we're going to do that before we get into protein synthesis. So as I mentioned before, DNA replication is just the process where an exact copy of a DNA molecule is produced. Okay. When this happens when two strands of DNA separate. Right? And when that happens, each is going to act as a template or pattern for the construction of its own complement. Now, this process generates new DNA-stranded, double-stranded molecules that are the exact replicas of the original DNA molecule. So we have the original parent DNA molecule here. We've got the two strands. They separate, and we have strand 1 and strand 2. And then we see where the second strand is synthesized here. Now, I want you to notice that you have the original DNA molecule, and that's called the parent. And then you can have the daughter molecules here, and those are just the molecules that are made during the replication. Now, this is considered semi-conservative replication. And when we talk about that, that's just the process where a new DNA molecule has one strand from the parent and a new complementary strand is produced. Now, replication occurs simultaneously at many points along a DNA molecule. So this is just happening pretty frequently. Now, let's talk about the specific steps here of DNA replication, because we've kind of looked at the overall view, and now let's talk about the individual steps and how this works. So we can view this as occurring in 3 steps. And the first step is the unwinding of the double helix. So we start kind of down here. This is the parent DNA, and the DNA unwinds, and we have a replication fork here. So the unwinding of the DNA, replication begins when DNA helicase, which is an enzyme, catalyzes the separation of the DNA double helix. Okay, now this unwinds the nucleic acid strands at specific points along the DNA helix. And in that process, hydrogen bonds between the complementary base pairs are broken, and the bases that were formerly in the center of the helix are exposed. Now, the point of this unwinding takes place, where it takes place is called the replication fork. So when we talk about the replication fork, right, this is the point the double helix unwinds during replication. right? And that's what's being exposed. So we've got the replication fork right here, okay? Now, in step 2, this is where we have the synthesis of DNA. And so DNA replication takes place along both nucleic acid strands that were separated in step 1. So we've got DNA replication happening here and happening here. Now the process proceeds from the 3' end to the 5' end of the exposed strand, which is the template, okay? So then, because these two strands are antiparallel, the synthesis of the new nucleic acid strands proceeds towards the replication fork, right here, right, and away from the replication fork this way on the other strand. And so the new daughter DNA strands form as nucleotides complementary to those exposed strands and then are linked together under the influence of the enzyme DNA polymerase. So let's put some words to that because I know that was a lot. So daughter strands form as nucleotides and are linked together under the influence of DNA polymerase. Okay. Then the second daughter strand is synthesized in a series of fragments. Okay. Now, when we talk about this, when we're talking, I want to point out this strand here is smooth, and then here we see a series of fragments. And then you also will see that there are what we call nicks, which are these gaps between the segments in the daughter strand. So nicks are just gaps between the fragments. Let's say the segments. I said fragments, but I like segments better. The segments in the daughter strand. Now, these segments, the DNA segments there, those are called Okazaki fragments, and they're named for the scientists who discovered them, which was a husband-wife pair, right? And these are just those DNA fragments that are produced during the replication. So that's those segments, and you can see they're pointed to here and here. Now, that gives us step 3, and step 3 here is where we've got to come in, well, not we, but the body has to come in and close these fragments here. And so we can just kind of sum that up as closing the nicks. And so what happens here is the Okazaki fragments are joined together by the enzyme DNA ligase. Right? And so that catalyzes that final step of DNA replication. And the result then is these two double helical molecules that are identical. Okay? So that's how DNA replicates. Now, let's talk about RNA. when we talk about RNA, it's helpful to compare to DNA. So this is a long, unbranched polymer consisting of nucleotides joined by 3 to 5 prime phosphodiester bonds. So, and you can see here's an example of a ribonucleic acid. So it's a long, unbranched polymer consisting of nucleotides joined by the 3' to 5' phosphodiester bonds. Okay. It can range from as small as 73 to as many as thousands of nucleotides. It's also generally found in the nucleus, cytoplasm, and the mitochondria. Okay. Some big kind of differences here that we see is that the sugar unit here is ribose rather than deoxyribose. Right? The other difference is that RNA contains uracil and no thymine. So it's got uracil instead of thymine. And then it's also single-stranded. So DNA is double-stranded and RNA is single-stranded, except in some viruses. But as far as we're concerned, we're going to say it is single-stranded. And that means that the RNA molecule doesn't need to contain those complementary bases in a ratio of 1:1. So now, it can, though, contain loops. So, for example, this is a loop of RNA where we do get this kind of looping structure there. Now, there's lots of different types of RNA. So what we're going to see here is and talk about is 3 types of RNA. We're going to talk about messenger RNA, ribosomal RNA, and transfer RNA. Now, each of these different types of RNA performs an important function in protein synthesis. Okay, so messenger RNA functions as a carrier of genetic information from the DNA of the cell nucleus directly to the cytoplasm where protein synthesis takes place. So messenger RNA carries genetic information from the DNA in the cell nucleus to the site of protein synthesis in the cytoplasm. Okay. And that's where in the cytoplasm, that's where protein synthesis takes place, right? Now, in contrast to DNA, which remains intact and unchanged throughout the life of a cell, messenger RNA has a really short lifetime. It's less than an hour. So it's got a short lifetime, less than 1 hour. And it's synthesized as needed and then rapidly degraded into its nucleotides. So it's synthesized as needed. So it's not just really hanging around in the cell. Okay. Now you can have ribosomal RNA, and this constitutes about 80 to 85% of the total RNA of the cell. Okay. And it's located in the cytoplasm in organelles called ribosomes. Right? And ribosomes themselves are about 65% ribosomal RNA, right? And 35% protein. Okay. And the function of ribosomes is really, that's the site of protein synthesis. Okay. Now, the third type of RNA that we're talking about is transfer RNA. Transfer RNA delivers amino acids to the site of protein synthesis. Now, here's an example of a transfer RNA. Cells contain at least one specific type of tRNA for each common amino acid found in proteins. Okay. Now, these tRNA molecules are the smallest of all the nucleic acids. They contain around 73 to 93 nucleotides per chain. So they're the smallest RNA with 73 to 93 nucleotides per chain. Right? Now, because of their small size, a number of tRNAs have been really studied extensively, and they have some kind of wild secondary structures. So this tRNA molecule has regions where there's hydrogen bonding between these complementary bases, and then we get loops where there are no hydrogen bonding. Okay. Now, two regions of tRNA molecules have important functions during protein synthesis. One of these regions is designated the anticodon, and that enables the tRNA to bind to the mRNA during protein synthesis. So the anticodon, which is this region right here, okay, enables the tRNA to bind to the mRNA during protein synthesis. Okay. Then the second important site is the 3' end of the molecule, which is shown right here. This binds to an amino acid and transports it to the site of protein synthesis. So we've also got a 3' end of the molecule. That's important. And this just binds an amino acid and transports it to the site of protein synthesis. Okay. Now, The reaction's catalyzed by an enzyme that matches tRNA molecules to their proper amino acids. And these enzymes, which are very specific for both the structure of the amino acid and the tRNA, rarely causes a bond to form between an amino acid and the wrong tRNA. Now, when a tRNA molecule is attached to its specific amino acid, it's said to be activated. So, when a tRNA molecule is attached to its specific amino acid, it is said to be activated. Okay, because it's kind of ready to participate in protein synthesis. This is showing right here is a general structure of an activated tRNA, where we've got that kind of 3' in there and it's attached to an amino acid here. So this is the amino acid and the 3' in is attached. And then this is showing you a schematic representation. And so where this is the whole big tRNA and this is just, again, the amino acid. And we're going to see that more in the next screencast when we're talking about protein synthesis. And so I wanted to point it out here before we got to that next screencast. So in the next screencast, we're going to look at protein synthesis. As always, if you have questions, please feel free to stop by my office.